Hi, my name is Nicholas Boulis. You all know Eric Perino, who is working with y'all to raise money for ALS. My understanding is that you know that Eric's dad, Mike Perino, has this horrible disease and is sort of actively deteriorating from it. The Perinos asked to talk to me about advanced therapies for Lou Gehrig's disease, or ALS, over the last year, given that they had received this diagnosis. And they really are an incredible family. One thing that we find with, well, that I've found with families of patients with ALS is they don't take no for an answer. And I think what you guys are involved with right now is tantamount to that situation, which is to say that they're not taking no for an answer. They're not just rolling over and perishing from this horrible disease, but actively trying to do something about it for their dad, for Mike, and for others with this disease. So they came and they spoke to me because I had pioneered a methodology for transplanting stem cells into the spinal cord. I'm going to use this whiteboard to talk to you. We initiated this trial in which we developed a device that allowed us to immobilize a frame to the human spine. That's a bad drawing of the spine. Inside the spine is something that we call the spinal cord. And inside the spinal cord, if we cut it longwise, what we find are cells and what we call white matter, which are the wires that connect our brain to our body. In patients with ALS, wires come down from the brain and they synapse on things called lower motor neurons. So those lower motor neurons then exit the spinal cord through what are called nerve rootlets and ultimately to go to muscle, and it moves muscle, okay? So what happens in ALS is these cells, these lower motor neurons, are dying, and the axons that connect the biological wires that we call axons connecting the cells to the muscle are dying back. So the cells are disconnecting from muscle and they're dying, and that effectively makes it impossible for patients to move. So over time, they slowly lose the ability to move and tragically generally die because they can't breathe or they can't swallow, which results in aspiration pneumonia. Patients have the option of undergoing a tracheostomy and going on a ventilator, but the large majority of patients decide not to do that because, one, it's incredibly expensive to maintain yourself in a state where you can't move at all, and number two, most people find the prospect of being completely paralyzed, I mean totally paralyzed, with the exception of your eyes, to be a horrible fate. So consequently, the family's taken up arms and is doing the best they can to raise money to develop experimental therapies. So what I did was I developed a methodology that allowed for safe injection of stem cells into the spinal cord, whereby those stem cells could protect the motor neurons. Together with Eva Feldman, who's at the University of Michigan, we executed a phase 1 and a phase 2 trial to test this, and those results are published, and I would invite you to look them up. They're promising. We showed that we could do these operations safely, and they suggest that these cells do protect motor neurons, but they're not a cure. The patients still succumb to the disease. Apropos to trying to develop other strategies, I had this discussion with the Perino family, and I said, well, one option is to access stem cell therapy. And since we've done phase 1 and phase 2 trials, one option would be using the right to try legislation or something called an expanded access program or a single patient treatment IND to try to get Mike access to these cells and to do this operation on him. And frankly, the company that manufactures these cells, Neuralstem, is amenable or open to the idea of doing single patient expanded access treatment or using the right to try legislation to try to access this. Nonetheless, at present, there's no easy way to do this because there is no ongoing trial. The reason for that is that to go to phase 3 was going to cost us about somewhere between $30 and $60 million, and we have not been able to find funding for that. Now, I don't expect your group to find funding for a $60 million trial. Having said that, we began to do a different thing for Mike, which is that there's a drug called Riluzole. Riluzole is an agent that is an anti-excitotoxic drug. So one thing that we've noticed over the years is that these motor neurons in the spinal cord, that they are surrounded by things called glia. Okay, I'm putting yellow in the glia. What these glia do, among the many things they do, is to take things called excitatory amino acids which are neurotransmitters that are used to excite motor neurons. So in order for this message to activate the motor neuron to send its message, an excitatory amino acid neurotransmitter is released. What that amino acid neurotransmitter does is it excites the cell. It stimulates it, which means the cell creates what's called an action potential. That action potential travels down the wire and activates muscle. The problem is, over years, we have noticed that in ALS patients, these glia cells are supposed to take up the excess excitatory amino acid. The problem is they have a deficit in a protein that allows them to absorb the excess excitatory amino acid. Consequently, the levels of excitatory amino acid are high in patients with ALS. All right? So, in order to address this problem, patients take this drug Riluzole, which blocks the receptors for excitatory amino acids. I really am a brain surgeon, even though I keep dropping this thing. So, this drug balances out the excess of excitatory amino acids that we believe is contributing to the death of these cells. And that makes sense, right? Because if you overexcite the cell, at some point, if it doesn't have the capacity to tolerate all that excitation, all those fluxes of ions going on in that cell to send these messages to the motor neurons, then they will die, and the ion cells will die back. So the drug protects the cells. The problem is that when you take this drug, okay, you take it at dose 1, okay, you get a little bit of a survival benefit. You go to dose 2, you get a slightly larger survival benefit. You go to dose 3, you get a slightly larger benefit. But what was found in these trials was at this dose, you begin to get liver toxicity. So, understandably, the FDA said you can't go past this dose because you're going to get liver toxicity. So, to solve that problem, what we did was we developed a preparation of Riluzole that you could put in a pump. Here's my little pump. I don't know if you think that looks like a pump or not, but there's a tube, and we put the tube going from the pump into the spinal canal. All right? We fill the pump with Riluzole. Okay? Now, garden variety Riluzole comes in a pill. You can't put a pill in a pump, so we invented a soluble version of Riluzole, and then we proved, using about $6 million, we proved when you pump this into pigs and dogs that it's safe. Now, apologies to the animal rights activists, but when you sacrifice these animals and you look at their spinal cords, and you take samples across the spinal cord, like this, what you find is the following. If you look at these different points, on oral Riluzole, you see this. So, no matter where you are in the spinal cord, if you're taking it orally, you have a certain amount. But the error's very large. That's an error bar. For those of you who don't know statistics, we're going to ask those of you who do know statistics to explain that. Okay, which means there's an average amount that is the same throughout the spinal cord. However, between animals, there's a large variation. The reason is, when you take a pill, the variance in absorption is great, and the levels vary over time. Now, if this drug is supposed to be acting to protect the neurons, what we want is for that drug to be consistent over time and consistent between patients. What the dog data shows is taking the pill is very inconsistent between patients and very inconsistent over time, okay? However, what we find when we put it in the pump is this. If the catheter tip is here, then what we see is that the levels are much higher near the catheter tip. And that the error is very small. So, more consistency between patients, more consistency over time, and you get a 10-fold improvement in the concentration of the drug. So, this is pretty much the only drug that's ever proven to keep ALS patients alive longer. If you take the pill, you get 3 months of extra survival. What you have to ask yourself is, would you take the pill and get 3 months, or do you want the pill and the pump, which gives you 10 times the amount of drug and gives it to you in consistent levels where it needs to act, okay? The only way we are going to prove that this is better is to do a clinical trial, okay? Right now, we don't have the money for a clinical trial. So what we're doing is we're just implanting it in people like Mike, and we're hoping for the best. And we're estimating safe drug levels based on our work in dogs. We've spent about, as I said, around somewhere between $6 to $8 million getting to this point. But we have to create a clinical trial. Part of getting to a clinical trial requires employing people to write what's called an investigational new device, an investigational drug exemption, investigational drug device exemption, investigational new drug, IND or IDE. These are regulatory approvals from the FDA that allow us to go ahead with clinical trials, all right? What we're trying to do is a phase 1 clinical trial, all right? Right now, Mike is being treated off-label, which means we're doing the best we can on the basis of limited information to try and keep him alive longer. I believe that this treatment can add time to his life, but it needs to have its time in court. It needs to have its day in court. That's a clinical trial. So to the extent that you guys are raising money, we need money to make this into a clinical trial. We need money to make this into a clinical trial so that we can get FDA approval for this, so that people's insurance companies will pay for it. Right now, Mike's family is paying for everything out of pocket. That doesn't mean he's paying me anything. It means he has to pay for the pump. His insurance company is not going to pay for that. It means he needs to pay for the drug, which is being custom manufactured. So they are spending a lot of money so that he can get access to this. Why? It's not because my team and my people are profiting. And in fact, we can't even create a for-profit entity that bills for this and then uses the money to create a trial, because people would say that we were conflicted. So the bottom line is, we need your generosity. We need you to raise money so that we can take this idea into a clinical trial. Whatever you're able to raise is going to be useful. That's all I got to say to you.